Hey guys, this is Colt Aiken, and I'm about to do my presentation over tight junctions and why they're important to the cells. Overview and general information of tight junctions are they are tight seals between two cells that are adjacent to each other and have proteins forming paracellular barriers and other functions. This is known as the kissing point of the cells, and you'll see that very clearly on the next slide. Tight junctions promote the use of diffusion, proteins, endocytosis, and exocytosis by making a single pathway, the apical side of the cell to the basolateral side of the cell. Structure and various proteins involved. There are three main families of tight junctions for protein families of tight junctions. And claudins are the most important, being that they're the major determinant of the barrier function of tight junctions, including the paracellular change and size selectivity that flows through. Occludins mostly do with signal transduction, as if we look over here, we can tell that this is like the fence function jams do. Then the barrier function is more the claudins, and then the signal transduction is more the occludins. And as you can see, these claudins bind to the ZO1 right here, this little protein or enzyme. Location and physiological role that tight junctions play are the location is the epithelial and endothelial cells located inside certain organs, and those organs are lungs, bladder, intestines, stomach, kidneys, just pretty important places. So this plays a huge role in our cells. Physiological role is it kind of acts as a multifunctional complex, and they coordinate a variety of signaling. They do lots of signaling. And trafficking molecules regulating cell differentiation and proliferation and polarity. These functions are really critical for epithelial and endothelial cell sheets to establish distant tissue compartments within the body and maintain homeostasis. Some of the diseases kind of associated with this malfunction of certain proteins. So if the tight junction barrier was kind of like messing up, like malfunction, your biggest subcategories would be cancer, inflammation, hereditary diseases, and vision loss and viral infection. As you can see on here, you get some of your common cancers that you see a lot. And just, and also Crohn's disease is probably the biggest inflammation one that you see. And cystic fibrosis is also on here too. One of my primary research articles I found was about what happens when you deplete ZO1 from the occludin or from the claudins protein, which if we go back is what kind of holds together these claudins and kind of enact, like it's what holds it's where the claudins kind of attach to the like actin cytoskeleton part. The ZL1 is the mediator between the claudins protein and then where the protein connects with the actin cytoskeleton. The results being that they found cells were visibly separated from each other. They used the atomic force microscopy imaging, revealed that only a few finger-like connections between the cells remain. The overall significant alternations of the cell morphology have only a small effect on the mechanical behavior of the individual cells that are still connected via adherens junctions. So overall, tight junctions are really important. If we come back over here, we can see that the tight junction is the first step in the junctional complex, and usually it allows endocytosis and it makes endocytosis and exocytosis happen through cells instead of trying to travel through this in between the cells. My question for y'all is, would a tight junction still be able to function properly if the gene for occludin is suppressed? And get back to me on that, and I appreciate y'all watching my presentation. Thank you.